Hello, everyone. My name is Johnny Lee, and the topic that I will be discussing today with all of you is targeting alpha-synuclein in Parkinson's disease. More specifically, the research question that I pursued along with my professor, Dr. Alf Detmer, was do small molecule inhibitors of protein aggregation also prevent its accumulation with lipids? And some of the key words that we used are alpha-synuclein, protein aggregation, Parkinson's disease, lipids, and cellular assay. Now, if you would like just to take a quick second just to read through our abstract that we wrote when we published our paper, I feel like this gives a very good idea of what our topic is going to be like in a very short paragraph format. I'll just give like a minute for everyone to read that. Okay, so now I'll be moving on. Um, so this is our table of contents. So today I'll be discussing an introduction, materials and methods, results, discussion and analysis, conclusion, and acknowledgements and some references at the end. So firstly, onto our introduction. So this diagram I actually borrowed from my professor. I feel like it sums up our topic very, very well. So essentially, many neurodegenerative diseases, including Alzheimer's, Huntington's, ALS, Parkinson's, they all share like a very common feature that's they have abnormal protein misfolding and aggregation. So these misfolded proteins form beta sheet oligomers and amyloid fibrillar aggregates, which contribute to the progressive neurodegeneration of neurons. So Parkinson's disease, in the case of my topic, is one example where the misfolded protein is alpha-synuclein. Now, what is Parkinson's disease? So, Parkinson's disease, or PD, is a neurodegenerative disorder that affects the nervous system. It typically leads to stiffness, slowed movement, and resting tremors. Other symptoms may also include balance issues and changes in speech or writing. The disease is associated with the loss of dopaminergic neurons in the nigra of the midbrain. So, a key pathological hallmark of Parkinson's disease is the formation of Lewy bodies. These aggregates are typically composed of, as stated before, misfolded alpha-synuclein. And this is also a diagram I borrowed from my professor. So as you can see, there's a significant difference between the control and the control brain and the Parkinson's disease affected brain, as well as you can see the aggregated alpha-synuclein as well. Now, so what is alpha-synuclein? Now, this is an underlying question that my professor and I first discussed when we began conducting this research. So essentially, early research suggested that Lewy bodies primarily consisted of beta sheet fibrillar alpha-synuclein. However, what my professor explored and what he noticed was that, and also these more recent studies have shown that these aggregates can also be lipid rich or membrane associated. In fact, some believe these lipid-rich forms may be more prevalent than previously thought. The exact role of each type in disease progression is still under debate, and evidence, however, suggests that both lipid-rich and fibrillar alpha-synuclein aggregates may contribute to pathology. So, as you can see in the diagram here, there's quite a difference between the alpha-synuclein aggregates in the membrane, which is the lipids, and in the fibrillar ones as well. Now, what is our study? Our study addresses a current treatment gap. So there are no approved therapies that target Parkinson's disease pathology directly. As we all know, there's no cure to Parkinson's yet. So most drug candidates here focus, however, most drug candidates focus on fibrillar alpha-synuclein, while lipid-rich aggregates remain typically understudied. So in our study here, we explored the therapeutic potential of small molecules, in this case, Fasudil, EGCG, and Li13B, all of which are known in the past by other studies that are known to disaggregate fibrillar alpha-synuclein. So, however, their effects on like lipid-rich aggregates, like these formed in this alpha-synuclein 3K model, which my professor developed, had not been tested. So essentially using live cell assay expressing alpha-synuclein. We treated these cells with these compounds and monitored changes in alpha-synuclein inclusion formation and protein levels. So essentially what we found was that Fasodil and Anli 13B, so I'll be showing this later in terms of diagrams and graphs, but these significantly reduce both inclusion formation and total alpha-synuclein protein. EGCG, on the other hand, had no noticeable effect. And another compound we used, ethanol, actually increased the formation of alpha-synuclein inclusions. So essentially, this study is the first to systematically test anti-aggregation compounds on lipid-rich alpha-synuclein inclusions. So essentially helping us understand how different forms of alpha-synuclein might be targeted in Parkinson's disease. So now on to our materials and methods. So firstly, for the study, we used M17D cells. They were supplemented with 10% fetal bovine serum, antibiotics, and L-glutamine. So to induce the alpha-synuclein YFP expression, we added doxycycline at 1 milligram per milliliter. So the compounds that we use, as I discussed later, was we are these listed here. So we tested several different compounds. So in previous research before, my professor discovered that trifluoperazine served as a very effective uh, inclusion, like reducing inclusion in alpha-synuclein. So what we essentially did was we used trifluoperazine as a positive control. And then the test compounds that we included to compare to trifluoperazine were EGCG, Fasodil, and Anli 138B, all which have previously shown to disaggregate fibrillar alpha-synuclein. So, and then ethanol was also tested as a solvent variable alongside DMSO. Now, for the drug treatment process, we conducted two assays. So, in the prevention assay, cells were treated with the compounds before inducing alpha-synuclein expression. And then in the clearance assay, so in the reduction assay, compounds were applied after the expression already been induced to essentially test their ability to reduce existing inclusions. And you can see the timelines here. Um, Now, for the imaging, what we used was an IncuSite machine. So the inclusion format was monitored every 4 hours using the IncuSite's live cell imaging system. Essentially, we use this image processing protocol to identify and quantify alpha-synuclein inclusions based on their size, intensity, and shape parameters. Now, on to our results. So essentially, in the study, like I said earlier, we used two different paradigms, so a prevention and a reduction. So in the prevention assay, cells were plated at 5,000 cells per well and pre-treated with compounds for 24 hours. The goal here was to see whether compounds could prevent inclusion formation from occurring in the first place. In the reduction assay, the sequence was reversed. We first allowed the inclusions to form by inducing the alpha-synuclein expression for 24 hours, and then only after the inclusions were developed, we were the cells treated with compounds to assess the drug's ability to clear and reduce preformed inclusions. So this was the statistical analysis format that we used. Um, it's the Kruskal-Wallis test. So each condition was tested with 36 other replicates. And then the asterisks represent different levels of significance. So yeah, as you can see here, these you'll see later on in our graphs what these essentially represent. Now, onto some of our actual data. So here you can see the representative YFP fluorescence images from the prevention assay. These images kind of visualize the extent of alpha-synuclein inclusion formation under various treatments. So as you can see, there's a DMSO vehicle control. There's the 8 micromolar trifluoperazine, Fasodil, Anli 138B, EGCG, and ethanol. Now, Inclusions here appear as bright green puncta, and it's clear that here that Fasodil and Anli 138B reduce these signals compared to DMSO and EGCG. So it's you can see here that these appear a lot dimmer than the DMSO, and there seems to be a lot less inclusions forming as well relative to the others. Now, on to the actual drug effects. So, in the prevention assay, we tested four concentrations of each compound. So, 0, 3, 10, and 30 micromolar. And we quantified inclusion formation, alpha-synuclein protein levels, and cell density. So, our positive control, which is 8 micromolar trifluoperazine, significantly reduced, as you can see here, it significantly reduced inclusion formation as expected. Um, when it came to our tested compounds, Fasodil and Anli 138B showed clear dose-dependent effects. So as you can see, the 3 micromolar had little to no effect. 10 micromolar reduced inclusions noticeably, and 30 micromolar nearly eliminated them. However, as you can see here also, EGCG did not inhibit inclusion formation at any dose, while ethanol surprisingly actually increased the number of inclusions. When measuring total green fluorescence as a proxy for alpha-synuclein protein levels, as you can see, both Fasodil and Anli 138B at 30 micromolar significantly reduced protein levels. EGCG, by contrast, increased total signal. In terms of cell density and cell viability, 30 micromolar Fasodil and Anli 138B both showed mild toxicity, but as you can see, 30 micromolar EGCG resulted in a clear cell loss. Now, moving on to our reduction assay. We so this is where again where we observe compounds whether they can clear existing alpha-synuclein inclusions. So these images reflect the treatments 24 hours after drugs were applied to already induced cells. The same concentrations were used as before. And as you can see, once again, notably, both Fasodil and Anli 138B again show visibly lower YFP signals compared to DMSO and EGCG, suggesting an active clearance of inclusions. In contrast, trifluoperazine seems almost ineffective in this context, surprisingly. Um, Now moving on, onto our reduction here. So in the reduction assay, we quantified inclusion signal intensity. Same thing as before, total alpha-synuclein protein levels and cell density, cell health. As we see previously, like triofluoroperazine, although it was effective in the prevention assay in reducing cell inclusion formation, it did not noticeably reduce existing inclusions this time in the reduction. However, Fasodil and Anli 138B, they both showed dose-dependent clearance effects. So as you can see, the 10 and 30 micromolar Fasodil significantly reduced inclusion levels, with 30 micromolar nearly eliminating them completely. Anli 138B kind of mirrored this effect with clear reductions at all tested concentrations, particularly at 10 and 30 micromolar. Once again, EGCG and ethanol showed no measurable effect, essentially confirming their ineffectiveness against clearing preformed inclusions. Looking at the total green signal, Fasodil and Anli, they both lowered alpha-synuclein protein levels, while trifluoperazine unexpectedly increased them. And then finally, we monitored cell density as a proxy for toxicity. So as you can see, 8 micromolar TFP and all the compounds at 30 micromolar, Fasodil, EGCG, and Li, they all showed signs of cytotoxicity, particularly EGCG, with a little drop there, as you can see. To capture the early drug effects, we also performed imaging 4 hours post-treatment. Even within this short window, Fasodil and Anli had already both reduced inclusion burden significantly. So this just further supports their potential as modulators for lipid-rich alpha-synuclein pathology. Now, onto our discussion here. So as we can see in our study, we tested 5 different compounds for their ability to reduce lipid-rich alpha-synuclein aggregates. Among these, we see that trifluoperazine TFP, Anli 138B, and Fasodil showed promising results. TFP primarily prevented the formation of inclusions without reducing total alpha-synuclein protein levels, whereas Fasodil and Anli not only prevented these inclusions but also reduced total alpha-synuclein levels. Um, these findings are significant because both Fasodil and Anli had previously shown efficiency and efficacy in disaggregating preformed alpha-synuclein fibrils. So here essentially we show that they also work on lipid-rich membrane-associated alpha-synuclein aggregates, which some argue to be more relevant, some argue fibrils. But either way, this suggests a broader mechanism of action. So these compounds essentially operate regardless of whether the alpha-synuclein pathology is fibrillar or lipid-rich. TFP seems to target aggregation and inclusion prevention, whereas we noted that Fasodil and Anli both may act as a dual mechanism, so they dissolve inclusions and also promote degradation. Um, this is particularly evident for Fasodil, which reduced both inclusions and total alpha-synuclein signal, even in short-term treatment. Um, Now, we next want to validate our findings. So essentially, it showed that the 30 micromolar treatments were consistently toxic. This just confirms that the high doses are not suitable and that 10 micromolar is a lot more appropriate, as it reduced both without toxicity. Um, Surprisingly, Anli was also effective at just 3 micromolar, so that indicates its potency even at low concentrations. And while we didn't perform IC50 calculations, the data suggests that 5 to 15 micromolar of either drug is sufficient. Um, we also see that both drugs show good potential for brain delivery. So Anli has shown to be able to penetrate the blood-brain barrier, and Fasodil not only penetrates but also is reported to enhance blood-brain barrier function and increase cell impedance. Both very desirable properties. Um, and when it comes to clinical considerations, high doses are associated with toxicity. So obviously, defining a safe therapeutic window is critical. Um, that being said, these compounds are very promising. So, for example, Fasodil is already an approved drug in some countries for cerebral vasospasm, and Anli 138B has already undergone animal testing for Parkinson's models with no significant toxicity. So essentially, the implications here are twofold. So first, these compounds could serve as starting points for alpha-synuclein targeting therapies, and second, their dual nature prevents inclusion and total alpha-synuclein. So this could be especially beneficial in diseases where both lipid-rich and fibrillar forms of alpha-synuclein coexist. However, more mechanical studies are needed to identify exact modes of action. Now, on to the future work here, hypothesis and clinical relevance. So, one important limitation of our study is that we did not explicitly measure alpha-synuclein redistribution, specifically the balance between cytosolic and membrane-associated alpha-synuclein. This could be addressed using sequential protein extraction Western blotting. So, this had previously been done before by Dr. Ulf Detmer in his lab. Um, our data suggests that reduction in inclusions may correlate with decreased membrane binding, but this also remains to be tested. We also did not assess the multimer to monomer ratio of alpha-synuclein, which is pretty important because monomeric alpha-synuclein accumulation is linked to neurotoxicity. So essentially, future studies should revolve around finding techniques to further investigate whether lipid-rich inclusions are more fibrillar than expected, explore whether these compounds can differentiate between beta sheet and lipid-rich aggregates, and examine degradation pathways. So whether these drugs act via some kind of mechanism. So now onto the conclusion. Um, it kind of just sums up everything that I've talked about before, but essential hypothesis kind of emerged from our research, which is that lipid-rich alpha-synuclein aggregates may be more fibrillar than previously believed, which could explain why Fasodil and Anli are both effective in dissolving them. Another is that reducing alpha-synuclein aggregation might indirectly promote degradation or clearance, so this could contribute to therapeutic benefit. Ultimately, in the end, understanding these is just the key to developing drugs that can safely and effectively reduce alpha-synuclein pathology in Parkinson's disease, and also related disorders. So now on to the final part, which is acknowledgements. I'll keep this brief. So I would personally like to thank Dr. Alf Detmer and his lab. So they not only allowed me to visit in person, but also served as a great mentor throughout the duration of this project. Thank you, CCIR, once again for helping do the connecting in between. So we were able to very fortunately successfully publish in February or March, I think. So this is the link to the journal. This is also the testimonial that I wrote for them. They provided great feedback overall. Some of these ideas on what to explore further were theirs, so that was also great. And yeah, so these are our references. Um, and if there are any questions, I would be happy to answer them now.
Okay, perfect. Thank you, Johnny, so much. That was a really great, thorough presentation. Are there any questions from the audience? You guys can raise your hand and then I can allow you to speak if you have any questions or even comments.
Hi, Johnny. Wolf Detmer here. Great job. Thank you for presenting our work. I'm now wondering, of the drugs you presented, which one do you think might have a real chance of being a therapeutic one day?
Um, so just based on what we had done, uh, and what I noticed overall, I feel like the obvious choice would be to say Fazodil, since it obviously did the most inclusion for me, reduction, but I feel like the toxicity issue with it is pretty prevalent. Um, so I feel like the safest choice to say which would be the most effective would probably be Anli. I'm not sure if you have done any further research with these, but yeah, if I were to make a bet, I feel like Anli would be the best just because of how potent it is in reducing inclusion formation as even in lower concentrations, such as the 3 micromolar, as we discovered.
Okay,
Yeah.
great. That's about it. I will leave Johnny as a panelist for a few more minutes in case anyone else thinks of any questions. And then in about 5 minutes, we will switch to our next presentation.
Okay, thank you very much.